semester?
No, this is the first one that we are going to do.
Okay.
And hopefully I have almost the entire semester like book, but it's difficult like to arrange the setup for the Zoom meetings, even if it's online. All the people is kind of taking courses or is pretty busy during the fall semester, so it's kind of hard. And I appreciate that you're participating.
Yeah, no problem.
I think we're live on the Facebook page now, so whenever we feel like is a good time to start, we're
Cool.
ready to go.
Okay, Colleen. I think that we are ready to start, Colleen.
Okay, great. Are you ready, Sarah?
Yeah.
Okay, well, I'll give you a quick introduction, and we'll do also a land acknowledgement for the campus, even though we're not on campus today. So I'll start with the land acknowledgement. We respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and the Beothuk. We would also like to recognize the Inuit of Nunavut and Nunatukevet and the Inuit Masinan and their ancestors as the original people of Labrador. We strive for respectful relationships with all the peoples of this province as we search for collective healing and true reconciliation and honoring this beautiful land together. So welcome everyone. This is our first OSC seminar of the semester. And today we'll have Dr. Sarah Lehnert, I hope I'm saying that correctly, talking on her presentation from the West Coast to the East Coast, answering evolutionary and applied questions in salmon using genomic tools. So Sarah, whenever you're ready, all you.
Okay, thank you. Yeah, so as Colleen mentioned, I'll be talking about evolutionary and applied questions in salmon, most of which I use genomic tools to address. But I'll talk about a few projects today that don't directly involve genomics as well. So to give you guys a little overview of my interests, I'm interested in the variation that exists in nature. And variation can occur at multiple different levels. For example, we have 8 million species or more on the planet, and these species look and act very different. But even within species, we can see variation where different populations might be adapted to different conditions, resulting in different populations looking really different from each other, even though they're the same species. And even within a population, we can have variation at the individual level, where different individuals can gain benefits by undertaking different strategies. And this individual level variation within populations, as well as between populations, is what a lot of my research focuses on. So I'm really interested in the mechanisms that act to maintain that variation in nature. And some of the work that I've done relates to the maintenance of alternative reproductive tactics, such as these male alternative tactics in salmon. I also work on color polymorphisms, and I'll talk a little bit about that work today. I'm also interested in the mechanisms that maintain immune gene diversity, especially genes like the major histocompatibility complex, and how that contributes to adaptation and mate choice in different systems. And also, most of my work focuses on salmonids, but I have dabbled a little bit in other species like invertebrates, working on the European green crab and how different invasion ecotypes can contribute to invasion dynamics. And more recently, a lot of my work has focused on larger genomic differences between individuals, like chromosomal differences, as well as more recently working on some variation that can be important for population persistence into the future. So I'll touch a little bit on some of these topics today. The first place I want to start is with some of the work that I did during my PhD, which my PhD thesis was entitled, "Why Are Salmon Red?" And this question of why are salmon red can be answered by saying because of carotenoids. Carotenoids are these red pigments that salmon get from their diet. They eat these pigments and then they deposit these pigments into their skin, eggs, and flesh. And this gives them those bright red colors that we know and love in salmon. Now, what's interesting about these pigments is they can be important for a lot of different functions within the body. So they can act as antioxidants and help the animal deal with stress. They can be important for supporting the immune function. They can help with vision, and they can be important when it comes to sexual selection. So there's lots of different reasons why a salmon would want to use carotenoid pigments, because they can have a lot of benefits. But what we know about a lot of fish species is that not a lot of fish do this. They don't deposit carotenoids into their tissues, especially not their muscle and flesh tissue like salmonids do. So this is a trait that's really unique to salmonid species. In fact, it's only found in about 4 genera of salmonids, where we get this red flesh pigmentation. But even though all these species exhibit this red flesh pigmentation, there's still a lot of variation that occurs among these different species and even within these species. And it's thought that it contributes to adaptation in some way. So we know that there are probably benefits to these carotenoid pigments, but because this trait hasn't evolved the same in all of these animals or even the majority of fishes, there are probably costs associated with carotenoids as well. So for my research, I was interested in trying to understand where salmon can gain benefits from carotenoids, but also where they might have a cost associated with them as well. So to do this, I worked on a Chinook salmon, which is a Pacific salmon species. I worked on this species because it's really unique among salmonids. It actually exists as a red phenotype and a white phenotype. Now, the red one is a normal type of salmon. It turns red during spawning on the outside. Its flesh or the inside of it is red, and its eggs are really red in color as well. But there's also this white morph that exists, and the white morph turns kind of a gray color during spawning. If you cut it open, it actually looks more like a cod. It has white flesh, and its eggs are really pale in color as well. So there's these large differences between them. Now, a lot of people assume that this is controlled by their diet, and it can be to some extent, where the more carotenoids a red salmon eats, the redder it can get. But a white salmon is genetically different from a red salmon. So even if it eats lots of carotenoids, it's going to stay white. It can never become red. So again, this trait is controlled by their genes. And what we know about these morphs is that they exist at different frequencies in different populations. Some populations are 100% red, some are 100% white, and some are 50-50. And a lot of my research took place in a river system where about 50% of the salmon are red and 50% are white. And this is in the Quesnel River system in British Columbia. So this system is located in part of the Fraser River system, which is a major salmon river in British Columbia. And this region is located about 800 kilometers or so upstream from the mouth of the Fraser River. So as I mentioned, I'm interested in trying to understand what the benefits are of carotenoids, but also what the costs could be. So for a lot of my research, I focus on the trade-offs that exist between red and white salmon. So we looked at this throughout multiple life stages, but today I'm going to focus on the early life stage of the egg stage. So during the egg stage, as I mentioned, we can see really big differences in the color of the eggs. So visibly to us, they look really different, like this picture here. But even though they look different, we can actually see measurable differences in the carotenoid content of these eggs as well, where the eggs of red females have about 6 times more carotenoids than the eggs of white females. So what we know about carotenoids in salmon eggs is that these carotenoids are supposed to improve incubation survival. And it's not just salmon that put carotenoids into their eggs, but a lot of other animals do as well. And if you had a chicken egg for breakfast this morning, then that yellow part of the egg yolk is actually full of carotenoids. So it's thought that a lot of animals do this because it can help protect the embryo during that period of rapid embryonic development, and that can help promote survival. So we might expect that the white eggs, because they don't have a lot of carotenoids, they might have lower survival during that incubation period. But there could also be a benefit to being a white salmon egg, and that's because maybe to predators, you're less visually noticeable. So it's possible that the red eggs can experience higher predation risk, but maybe they also survive better in the gravel. So there could be these trade-offs that exist. So for this work, we used a common garden experiment where we created crosses of salmon and followed them from the egg stage to later stages of juvenile development. And I'll focus on some of that earlier life stages today. Just to situate you in this experiment, this is what it looks like in the Quesnel River where we catch the salmon. We set a net across the river using a boat. That net then drifts to shore. The water's fast and deep here where the salmon are getting ready to spawn. Once the salmon are tangled in the net and they're brought to shore, we can untangle them. We can start capturing them to bring them back to the research station. And then we hold them under semi-natural conditions so they can start to get ready for spawning. Once they're ready for spawning, we can collect gametes, such as the eggs and sperm. We combine those to make different families of fish. And then we can rear the eggs in incubation trays and continue to follow them throughout their eggs stage, as well as into later stages of development. So the first question that I was interested in for these eggs is there differences in the survival of families of red eggs versus white eggs? And what I found here was that on average, there was no significant difference in egg survival between red salmon and white salmon. So although we expected that carotenoids should provide them with a benefit in terms of survival, we did not see that. But another question I had was, well, we know how much carotenoids are in the eggs. So if we look at within these two morphs, do we see differences in terms of carotenoids and survival? And what we found was really interesting. Within the red Chinook salmon morph, we saw that as you increase egg carotenoid content, you also see higher survival in those families. So there are benefits to having more carotenoids in your eggs, but we only see this being true within the red phenotype. If we look at the white salmon, there is less variation, but even ones that had the lowest level of carotenoids still did really well in our experiment. So overall, this suggests that in the white morph, there's not a disadvantage to having less carotenoids, but there is in the red morph. To us, this suggested that maybe white salmon have evolved some sort of mechanism to compensate for their lack of carotenoids, that they could potentially add other antioxidants to their eggs to help increase incubation survival when they don't have a lot of carotenoids in their eggs. So this is one hypothesis that we have. Now, it's not just egg incubation survival that's going to be important, it's also how those eggs are interacting with predators. So if we look at a spawning salmon like this female here, she's getting ready to spawn. There's some male salmon around, but there's also some other species like rainbow trout. Once the female starts to lay her eggs, those rainbow trout will actually start to feed on those eggs. And it's not just trout, but it can be other species of salmonids and other fish that often eat salmon eggs. So I was interested in using rainbow trout as an ecologically relevant predator to see if they show higher predation risk on red eggs versus white eggs. So to do this, I used a behavioral experiment where we set up video recordings of trout in tanks, and then I released a red and a white egg into the tanks simultaneously. When I did this, I recorded what egg was eaten first, the red one or the white one, and I also recorded how long it took for those trout to eat the eggs. In this case, we released lots of eggs over multiple different trials over multiple days to see what patterns would emerge. And in this experiment, we actually used two different populations of rainbow trout. One population had prior experiences with Chinook salmon eggs because their population is located close to that Quesnel River system, whereas the other population would have had no experience with Chinook salmon eggs. So we thought maybe they could show some differences because of those backgrounds. But what we found in this experiment when we looked at which egg was consumed first is that in both populations, red eggs were consumed first twice as often as white eggs. So there was a significantly greater predation risk on red eggs. If we looked at the time it took for those eggs to be consumed, we also saw that red eggs were eaten significantly faster than white eggs in our experiment, again suggesting that red eggs have a higher predation risk on them than white eggs. So through these experiments, we were able to detect some of these trade-offs that are associated with carotenoids. At this egg stage, we see that within the red morph, there are benefits to having more carotenoids. More carotenoids leads to higher incubation survival. But having more carotenoids can also lead to greater predation risk. So there are these trade-offs that are associated with these carotenoid pigments. Now, in addition to the egg stage, we looked at multiple other stages in these salmon, and I won't have time to get into results from all those experiments today, but I want to talk a little bit about what we found overall. So after these eggs hatched and started to develop, we monitored the growth and survival of those juveniles, but we didn't see any difference between salmon that came from red eggs or white eggs. We also started to look at their immune function by challenging them with a virus and then looking at their response to that virus using gene expression. But we didn't see any response difference between red and white salmon in that experiment. So although carotenoids are supposed to be important for immune function, we didn't see that they were in this case. But another thing that we did find is that there are significant differences in the immune genes of red and white salmon. They have different levels of diversity and different alleles, which maybe could be a way that white salmon could compensate for their lack of carotenoids. Maybe they can fight off infection through having these different genes that are more, or alleles that are more diverse. And finally, we also looked at the adult stage in these fish. So when I started this experiment, I thought maybe these fish mate assortatively and based on their color, and that's how this is maintained. But we found that these fish do interbreed in the population, but there is a bit of assortative mating happening, where red and white females often bias the paternity of their eggs in favor of a male that's the same color as themselves. So they can do this through different mechanisms like pre and post-copulatory processes. And we've measured this in both the red females and the white females. So that's just a quick overview of some of that work that I did during my PhD, where I think this system is really interesting from an evolutionary perspective. But it's not just interesting for evolution. This trait of pigmentation is also really important economically. And that's because consumers such as ourselves will pay more money for a redder salmon. So aquaculture producers that make salmon, they often want to have salmon that are redder in color. So they can do this by adding more carotenoids to the diet, but that doesn't always work. So carotenoids are expensive. And even if you feed your salmon a lot of carotenoids, you can still have variation that exists between individuals. You could potentially selectively breed for this trait, but it's kind of difficult because once the fish reach sexual maturation, they actually start to lose their carotenoid pigments because they start to move it out of the flesh. So it's hard to see those differences at that late stage in development. So instead, we could identify the genes that are important for color, and then we could create some marker-assisted selection programs where we breed salmon based on their genetics. So I was interested in trying to find the genes that make a salmon red. What we wanted to do first is create a family of fish that has a variety of colors in it. So to do this, we took a salmon that is red and a salmon that is white from different populations, and we crossed them together to produce F1 offspring that would carry the red alleles and the white alleles for color. Then that fish was back crossed to a white salmon. And what this does is it allows recombination to happen. And what you end up getting is a family of fish that shows a variety of colors from really red to really white. So with all these fish, we then looked at a color score for each one of them. So we said this fish is a 1 or this is a 5 based on how red or how white it was. And we genotyped all these fish using thousands of markers across the genome. With this, what our goal was to look at each genetic marker and see if any of them were associated with the color patterns that we saw. So this is a method called a genome-wide association study, or GWAS. And this is what we found when we did the experiment. So in this figure here, this is called a Manhattan plot. Along the x-axis, you have the chromosomes of the salmon, and the dots represent each genetic marker in our study. And they're positioned along the chromosome based on their genetic position. On the y-axis, you have the significance of each genetic marker. So ones that have higher values are more significant. So they're more important for explaining the differences we see in color. What you can see, hopefully from this figure, is that there's a whole bunch of dots on chromosome 30 here in blue that are really significant. These ones are all above this red line. And the most important marker here is at the top of this graph, which is on chromosome 30. And when we look at the genotype and the phenotype at this genetic marker, what we see is that individuals with different genotypes show significant differences in color. So ones that have this GG genotype, they're really pale or white in color, whereas the ones that have this other genotype, GT, are really red or darker in color. And this specific marker can actually explain about 66% of the variation in color in these salmon. So it's likely that this marker is located close to the gene that controls color in salmon. So that brings us to the next question. Well, what gene is closest to this genetic marker? And when we looked at this, we found that the gene that was located beside this genetic marker is one that's called beta-carotene oxygenase 2-like gene, or BCO2. BCO2 is actually involved in the cleaving of carotenoids, so it has a direct function related to carotenoid metabolism. And it's also found in other species like mammals, birds, and reptiles as being important for explaining carotenoid accumulation. So this was a really cool candidate and a promising gene that probably influences color in salmon. And we looked at it a bit further, and what we find is that, so this gene encodes an enzyme that is responsible for breaking down carotenoids. So what we find is that in white fish or in white salmon, it's actually the expression of this gene that makes them white. So when this gene is operating properly and being expressed, this enzyme is breaking down carotenoids into colorless derivatives. So then they don't accumulate as red pigments, and instead the animal is white. But if we disrupt this gene and stop its function or reduce the expression of it, what we find is that these fish will start to accumulate those carotenoids because they're not being broken down. And that will make the fish have this red color in appearance. So through the genetic and gene expression work that we did, we could determine that this was the reason that we have these red and white salmon. So coming back to this question of why are salmon red? Well, the easiest answer is that it's because of this mutation that happened in this BCO2 gene. And this probably happened in the common ancestor of salmonids, which happened probably about 30 million years ago when these different species were diverging. This mutation reduced the function of this gene, allowing carotenoids to accumulate in the muscle tissue. And this was potentially important for leading to some of the other life history variation that we see in salmon, like the evolution of anadromy. But it's only within Chinook salmon in Oncorhynchus that we see that a white Chinook salmon morph has evolved through another mutation increasing the function of this BCO2 gene. This probably provided these salmon with an advantage when it came to predation risk, allowing this morph to exist and persist in certain river systems on the West Coast. So that's an overview of some of the work that I did when I was doing my PhD. But after I finished my PhD, I moved out to the East Coast to Halifax, and I started working in the Bradbury Genomics lab. And then when I was working there, I was mostly working on Atlantic salmon. So I want to talk now about some of that work. And I know a lot of you might know about Atlantic salmon, but I'll give you a quick overview of them anyways. They are found across the North Atlantic Ocean, and they occupy river systems in Europe as well as in North America. So although salmon from Europe and North America are considered the same species, they actually diverged a really long time ago, up to 600,000 years ago or more. And even though they're the same species, they do show differences in the number of chromosomes. European salmon have 29 chromosomes, but salmon here in North America only have 27. And that's because of some rearrangements that have happened between the chromosomes. And I'll talk more about these rearrangements today. Now, these salmon have been isolated for a long time. They have occupied different glacial refugium during periods of glacial maximum. But even though they've spent a lot of time isolated from each other and are genetically very different, they have had some opportunity for contact. So some earlier work using mitochondrial markers has shown that in some places in North America and in Europe, salmon have crossed the ocean and interbred. So if we look at a map of these mitochondrial haplotypes in these pie charts here, the green ones represent North America and the red ones represent Europe. If we look at Europe over here on the map, most of the fish have this European mitochondrial haplotype. So they have this European lineage, except for some places in the north in Northern Europe. If we come over to North America or Canada, we see that most of these fish down here in the maritime regions are this green color, which is North American type. But if we look to Newfoundland and Labrador, we actually start to see more of this European lineage showing up. This is especially true for here along the Avalon. A lot of the populations here seem to have a high proportion of European mitochondrial haplotypes. This suggests that salmon from Europe crossed the ocean and interbred with salmon here in North America, and those signals are still present in their mitochondrial DNA today. So what I'm interested in is how this might persist in the rest of the genome of Atlantic salmon. So although we have this evidence from these mitochondrial markers, there's no evidence yet that this has influenced the nuclear genome of a salmon. So I was interested to know if these contact events have had introgression of European DNA into salmon DNA here in North America. And I should mention again that this isn't something that has happened recently. It's expected that this happened about 10,000 years ago. So anything that is still present in the DNA of salmon today might be important and under selection if we can still detect it at this time. So for this project, what we did is we looked at high density genomic markers, so spread across the genome for salmon populations in North America and in Europe. In Europe, a lot of our populations are centered around Norway, and in North America, we have populations from New Brunswick and Nova Scotia and Newfoundland and Labrador. So first, what we did is we focused on these North American samples, and what I wanted to do is find areas of the genome that were differentiated between populations, but also highly differentiated between individuals. And when we did this, we used a program called PC Adapt. This is PCA based. And what it does is it helps find the markers that are important for explaining differences either between populations or between the individuals. And again, this is a Manhattan plot where higher values for the markers indicate that they are more significant. When we looked at this, what we found was that there were 4 big peaks on these chromosomes, and these were chromosomes 1, 8, 23, and 29. And what's really interesting about these chromosomes is that they're actually involved in what we call chromosomal rearrangements between European and between North American salmon. So chromosome 1 and 23 are involved in a translocation in North America, and 8 and 29 are involved in chromosomal fusion in North America. So you might be wondering what I mean by this. Well, I'll try to explain it in a bit more detail. So there are different rearrangements that you can have in a chromosome that result in large structural changes to those chromosomes. And one of those can be a translocation. This is when one arm of a chromosome breaks off from another one and attaches to a different chromosome. Another thing you can have is a chromosomal fusion. This is when you have 2 chromosomes that are separate, but then they come together and fuse into a single chromosome. And then another type of rearrangement that I haven't mentioned yet is called an inversion. And inversions are these large chunks of the chromosome that get flipped around and reordered. So for example, here you might have one order of the genes, but another another individual might have the opposite order. So those genes are reversed, and that is considered to be inverted. So that is an inversion. And the reason that I bring up all these different rearrangements is because they have recently been found in a lot of different species, and they can lead to these things called supergenes. Because the chromosome structure is different between different individuals, this can result in less recombination in these regions. And what that allows is for these alleles within those regions to start to co-evolve together into tightly linked complexes. So what I mean by this is that within these super genes or these regions of the genome, you can start seeing the genes that relate to different functions starting to work together and evolving together. So for example, you might have genes that relate to behavior and color and reproduction. And what this can do is create a complex of genes that are important for contributing to a complex adaptive phenotype. So for example, we often see that these super genes that are due to rearrangements can lead to adaptations like ecotactic or color. These tactics, they look different, they act different during reproduction, and this is due to an inversion that acts as a super gene. So doesn't only occur in species like these birds, but it can also happen in other animals. So we see that inversions are important for Atlantic cod, for contributing to ecotypes. We also see that they're important in Oncorhynchus mycus, which is the rainbow trout, but also the steelhead. So inversions make the difference between it migrating to the ocean versus it staying in the river. We see the same thing in herring populations where supergenes are important for contributing to spawning ecotypes. As well, we see this in capelin. So there's lots of examples of these rearrangements being important for adaptation. Now, to bring it back to Atlantic salmon, when we started this project, we knew that these rearrangements were different between Europe and North America. But what this analysis suggests, which is just done within North America, is that there's actually chromosome differences within North American populations. And we didn't know that these rearrangements were involved in these chromosomal differences before. And it's not just different between different populations, but it's actually different between individuals within the same rivers here in North America. So the next question that I had was I wanted to understand why this evolved or how it came to be and why it continues to persist. So what we did next was we looked at the genetic relationships in these parts of the genome. First, we looked at the genetic relationships that exist on average if you looked across the whole genome of the salmon, and we saw a pattern that we would expect to see, where all the salmon from North America grouped together. So this is a neighbor joining tree where each branch is representing a different individual here. And we see that all the North American fish are found in this part of the tree over here, whereas there's a long branch before you get over here to Europe. So showing that European and North American salmon are genetically different, and these fish generally group by geography within North America. But if we then look at these different parts of the genome, like where these rearrangements occur, what we found was that the trees that we create are actually quite different. And I won't get you to focus on all the details of this tree, of these two trees, but I will say that it helped us understand how this variation arose and why it is present in these populations. So I'll talk about that in a different diagram than trying to explain these little trees here. So first, if we look at this chromosome 8 and 29 fusion, what we know about this is that in Europe, it's two separate chromosomes. All individuals have separate chromosomes for 8 and 29. In North America, you have this type as well, where there's individuals that have two separate chromosomes, but there's also individuals that carry these fused chromosomes as well. So two different chromosome types exist in North America. And some individuals even carry a copy of the fused and a copy of the non-fused type. Now, what we found is that these two types actually exist in all the populations that we sampled in North America. So that tells us that this was present before the end of the last glaciation, and it has spread through all the populations as salmon recolonized their range after the end of the last glaciation. So this is an older polymorphism that existed and has spread through all the populations. But we found a very different story when we looked at chromosome 1 and 23. What we know is that in Europe, the standard type is to have chromosome 1 and 23 as separate chromosomes, so no translocation. But in North America, the standard type was thought to actually be the translocation, where you have one arm of one and the other part of one attached to chromosome 23. So in this case, this is the old standard type that we see in North America, and it has been present for a long time, and it has spread through all the populations as salmon recolonize their range. But what we also found is that some populations have this European type. So that European type was introduced into North America, and it was introduced only into some populations, and that type remains today. And within these populations, we see that they can carry either this translocated or non-translocated type of chromosome. So there's that individual variation that we see between within river systems due to that introgression from European salmon that happened about 10,000 years ago. And most of this is located in Newfoundland as well as in Labrador. So hopefully you followed all of this stuff about the different chromosomes, but if you didn't, what I'm basically saying is that through this genomic work, we were able to determine that salmon within rivers in North America can have different numbers of chromosomes and different arrangements of their chromosomes. We did know this from earlier work that there had been suggestions based on karyotyping work that there was variation in chromosome number within salmon rivers, but we didn't know what chromosomes were involved or why that might have evolved. Whereas this work through genomics tells us that these two chromosome types have evolved through different evolutionary histories, one through polymorphism, and one has been introduced through secondary contact. So this has been really interesting. And the reason, again, that we care about these rearrangements is because they can start to act as supergenes that can be associated with important life history characteristics or environmental variation. And some more recent work on these rearrangements has found that indeed they are associated with environmental characteristics in salmon populations. And our work has shown that they're under selection. So here that we can show why they might be advantageous and why they might persist in a lot of these populations. So again, I just want to reiterate how important these rearrangements can be and that they don't only apply to salmon or fishes, but they can be found in all these species pictured here, as well as in different birds and fishes and plants and invertebrates and mammals. So there's lots of animals and plants that have these rearrangements that are important for adaptive phenotypes. So if you study a complex phenotype, maybe you should consider if something like a chromosome rearrangement is contributing to that adaptation. So that's a little bit about the history of Atlantic salmon and their genetic legacy, but now I want to focus on the future of Atlantic salmon and talk about some work that I'm doing right now. I'm starting to not have too much time left, so this won't take very long, and I'll just kind of highlight some of the work that I've done recently and some preliminary work that's going on. So some of the work that I've done recently has looked at declines of Atlantic salmon populations. And using genomic tools, we can estimate what populations of salmon have experienced declines. And what we're finding through my research is that about 60% of salmon populations have experienced significant declines in recent decades. Now, there are a number of factors that can contribute to these declines, and overall, we don't really fully understand why salmon populations are declining, but we know there are many factors involved. And one of those key factors is thought to be climate. So it's important that we start to consider how salmon might have to be changing in response to climate change. So I'm interested in using genomic methods to predict how salmon will respond to climate change. And this type of work is being done in other species. And to do this, what we need to do is we look at how the genetic variation that exists today contributes to adaptation to climate variation. So we look at the associations that exist between the genotype and the environment, and then we can start to look at the future environment to understand how much populations will need to change in the future. This is a measure that we call genetic offset, or sometimes called genomic vulnerability. And basically what this measure will tell us is what populations are most vulnerable to climate change. And this is determined by which populations will need to undergo the greatest shift in allele frequency to match future conditions. So what populations will need to change the most genetically to adapt to future environments. This type of work has been done in species like the yellow warbler, where they've identified populations that are most vulnerable. And these populations, it turned out, were ones that had already started to experience significant declines. So there is an association between this future potential vulnerability and ones that had already been starting to show negative consequences. Other work like this has been done through the OFI with the species of Arctic char, looking at how char in Labrador will be vulnerable in terms of climate change. So I'm interested in this from the Atlantic salmon perspective. So for this project, we use high density genomic data where we have almost 4,000 individuals that have been genotyped across almost 100 locations in Eastern North America. And with these samples, we then look at what part of the genome is associated with the current climate conditions. And we can do this by using climate data that's available online through the database of WorldClim. And with this, we can identify what genes or what markers are associated with the current climate conditions. Then we can look to the future climate conditions, and this can also be downloaded from the WorldClim database, where for the year 2050, we can download these bioclimatic variables and start to look at how salmon will need to respond to match future conditions. Now, again, this is a measure called genetic offset, and we've done this for all salmon rivers in Canada by using these current associations to model what they would need to look like in the future. So through this work, what we find is that if we look and project these metrics into the future for over 900 salmon rivers in Canada, we can see that in the year 2050, some populations will be more vulnerable than others. And those are ones that are in this map here with the bigger circles that are darker red or darker orange in color. Those are ones that will be most vulnerable. These ones are often located in the mid portion of the range, like Quebec and Newfoundland. And this is actually where, if you look at the genetic signals, the slope is steeper. So there's a steeper environmental gradient. So even when the environment changes by the same amount, the salmon in that portion of the range will need to change more genetically to match those future conditions. There's also populations outside of that range, such as in southern Nova Scotia and in central Labrador, that also show high vulnerability. So this is some preliminary analysis that shows how we can start to integrate genomic and environmental data to help understand changes to climate, and this can help inform management strategies. So here, this is just one part of the response that we can start to predict how salmon might need to change in the future. So to summarize and conclude today, I hope that I've convinced you that these genomic tools can help us address some important evolutionary and applied questions in a lot of species. We can see how these methods can be used to identify mechanisms that help maintain variation in nature. We can see what genes or what chromosomal rearrangements might contribute to important adaptation in these species. And genomics can also help us understand how populations have changed historically, but also help to understand how they might be changing into the future as well. So with that, I'd just like to thank everyone for listening and thank everybody who's helped me with these projects over the years. And I would be happy to take any questions now, or if you want to email me later, that's good too. So hopefully you enjoyed this talk today.
Thank you so much. Does anybody have any questions? You can either put them in the chat box, or if you're in the Zoom meeting, you can unmute yourself and ask the question yourself. Okay, well, it seems like there might not be any questions. So thank you so much. Oh, okay. Yeah,
I have
go
some
ahead.
question for you, Sarah. I would like to say thank you for your great talk. It's a really nice work, years of work. And I know that you were saying that there was some difference between the Chinook salmon, Chinook salmon, sorry. And do you know if it those difference, because you say that someone were red and then the other ones were white, and those also obviously probably are due to knockdown of genes or SNPs. Do you believe that these SNPs are more focused on the inbreeding of the animal, or are
So I don't think it relates to inbreeding at all. I guess in some populations you have red ones only, in other populations you have white only, and then some populations you have equal amounts. So it seems to depend on the environmental conditions that they encounter for where these types can continue to exist. But I guess there could be some founder effects in some rivers if they happen to be colonized by mostly white fish, and maybe there's an advantage to being white in that population, then I guess that could contribute to what type is present there now. But I don't think one of those types is more susceptible or has higher levels of inbreeding.
Sarah, I have Ian here. I have a question for you. I was just trying to understand, I was really interested about your predictions in relation to future climate change and the populations that would be most susceptible. Mm-hmm. And I was just trying to balance that with the observation that it's not the populations in the middle of the range that have been disappearing, but it's the populations on the southern areas of the range that have been most affected, which I guess we would expect. But I'm sort of trying to align with what you're predicting from the amount of change that has to occur, and that those populations in the middle area presumably would be the ones that would experience the greatest declines, but we're not necessarily seeing that.
Yeah, for sure. And it puzzled us at first as well. So I will say that this is just one component of it. So this is just the climate perspective, whereas I think there are lots of other things impacting salmon, especially in the southern portions of their range. But I guess based on the genomic data, what we're seeing is that ones that exist now in warmer areas should be well adapted to warmer conditions, whereas ones in the north should be adapted to colder conditions. And the ones that are in the middle are ones that will have to undergo greater genetic change, even if the environment just changes by the same amount in those populations as it does in the south or the north, just because of the steepness of decline in that area. But yeah, it's definitely something that we're trying to figure out. And I think it doesn't encompass everything that we can look at. It's just kind of one part of the story, but I think there are other aspects that we can definitely look at, like plasticity and the potential for mutation in these populations that could potentially give them advantages or disadvantages under climate change.
Thanks.
I had one question. Thank you, Sarah. Excellent talk. For the referring to the BCO2 work, are there SNPs that are of interest in industry for selecting broodstock that would have a darker red flesh color, and can the Chinook information be kind of transferred into other salmonids for kind of identifying genes that might be useful in selecting flesh color?
Yeah, so I think when we started this project, I don't think we knew what we were going to find, that it was really a major locus that controlled these differences in color. So BCO2 seems to be important for kind of allowing a salmon to be red versus white. But some of the other work that's coming out of some of the labs in Norway are showing that some of the variation within red is controlled by another BCO gene, BCO1. So maybe if you want to have a red salmon, you need BCO2 to be non-functional, but then you can also change the expression of BCO1 to make that salmon even redder. So based on the work of my work and that work, I think that it's a combination of a few different genes that we could start to selectively breed salmon to get them as red as possible.
That's great, thank you.
I apologize because my Zoom just go off, but I'm sorry, that's things that happen. I don't know if anyone else has questions. No. So I have another question, but based on the Atlantic salmon, and was very interesting to see that you have rearrangement regions that jumps into one chromosome to another, or do the translocation or the fusion. Do you think that this fusion or the rearrangements are due to the crossover between European and North America, or is really conserved between the population that is in North America more than a cross between the different countries' populations?
Well, so the chromosomes are different between Europe and North America, but they are variable within populations here in Newfoundland. So I'm not sure I understand your question, actually, sorry.
Well, the thing is that this rearrangement regions, right, supposed to be like trigger for promoter regions, or I don't know, transposases or any other genomically genomic elements that trigger that the chromosome jumps from one to another. Do you think that these rearrangements are conserved between the populations, like between North American populations rather than the European, or are triggered by they cross each other?
Yeah, they're not created because of created because they're crossing together, I guess. So in North America, it looks like the fusion exists because some individuals have it and some don't, and it might be an unstable part of the genome that within North America, we only see variation in it, whereas in Europe, there's only one type that exists. They're definitely not fused. So I'm not sure if I've answered your question still.
Yeah, I imagine that are like conserved between the North American population and the European. But did you, I don't know if you did an analysis of like a locus like these rearrangement zones, like to see how concerned they are?
So I guess I'm not sure if I know what you're asking, but we see that like in some populations, lots of fish have one type of the arrangement and lots of fish have the other type. But what do you mean by conserved, I guess?
Like if you do that you were looking for the SNPs, right, between the populations to see if these SNPs were specific for one population, like within the North American region. I don't know if you did an alignment of those region that have the SNPs. That
Okay.
was my question.
Yeah, so I guess the type for what the translocation is so the type that is European that exists in Canada, it looks exactly like the European type that's in Europe. So it hasn't really changed much. But for the fusion, there are starting to be differences between the ones that have the same chromosome type for North America and in Europe. So it's not as conserved as we see with the other rearrangement.
It's good. I think that is all my questions. I'm pretty sure that I don't know if anyone else have another question. You can put it in the chat or just open your mic and make it public. So, okay, I think that's everything. And I really would like to say thank you, Sarah, for your time, for your talk. It's very interesting. And well, Matt will be glad to have it in his course. And
Thank you.
and thank you for share this knowledge with us. And that's it for today, I guess.
Thanks for giving me this opportunity. Yeah, that's good.
Bye. Thank you.